presentation, which is 8 minutes only, and then followed by Q&A, questions and answer for 2 minutes. Dr. Lina will actually tell us when the time's up. So student must stop here. So the first presenter today is Nur Irdina, and then followed by Amirah Shazina. Then the third presenter is Amirul. So at least these 3 students can stand by. Okay, so without further ado, I would like to invite the first presenter, Nur Irdina.
Hello, Assalamualaikum, Doctor. Okay. I will share my slide. Doctor, can you see my slide?
Okay, clearly.
All right. Can I start?
Yes.
Okay, Bismillahirrahmanirrahim. I'll start first, okay? Okay. Bismillahirrahmanirrahim. Wait for a minute. Okay. Bismillahirrahmanirrahim. Hello, Assalamualaikum to all the audience, and I greet to all the fellow examiners. Today, I would like to present my final year project. My name is Nur Irdina binti Ismail, a biology student. I would like to present my FYP, which is entitled Antimicrobial Effects of Allium Sativum Garlic Extract. The first slide is introduction. Allium sativum or garlic is one most best-selling and studied herbal remedies and widely used to treat different health problems. And the example of medicinal application such as combating cardiac arrest, cough, and a digestive problem. As STI or skin and soft tissue infections contributed from a microbial invention, which commonly involve Staphylococcus aureus and Streptococcus pyogenes. Next, there's 2 types of the Allium sativum, which is artichoke softneck and the silver skin softneck. The characteristic of the Allium sativum are the bulb grows up to the 4 feet in height and contain a colorless liquid compound called allicin. The garlic leaf is compressed and elongated, and the allicin compound can be obtained by the squeezing or slice the garlic clove. And it also provides a pungent taste along with a sufficient nutritional benefit. Next, the problem statement. The problem of this study is arising the number of antimicrobial resistance against bacteria and fungi due to the lack of consistent in taking an antibiotic. The challenge to treat the skin infections using a synthetic antibiotic, which may cause a long-term of the side effect to human. The significance of the study increased the awareness of the medicinal importance of the Allium sativum or garlic as an antimicrobial agent in curing the antimicrobial infections which are used in the daily life and more cheaper compared to the synthetic antibiotic. Second is objective. The objective of this study to determine the antimicrobial activity of the garlic extract against a selected bacterial and a fungal skin pathogens. Next is a flowchart. The procedure of this experiment, including the preparation of the garlic extract and the preparation of the media and broth. After that, culture and the subculture of the microorganism. After the subculture the microorganism will diffuse into the Mueller-Hilton agar using a distribution method. And the last one is the data analysis. Fourth is the result and recommendation. This is the table of the mean of diameter zone inhibition of a garlic aqueous extract. As you can see, the 3 different extract concentrations indicate to have a zone inhibition against Staphylococcus aureus. However, E. coli show zone inhibition in the extract concentration of 100% and 75%, but the negative result can be observed through zone inhibition indicate the low resistance of the candidate albicans. Next, the table of the mean of diameter zone inhibition of a garlic ethanolic extract. All the three extract concentrations showing inhibition zone indicate as a sensitive response against the Staphylococcus aureus and E. coli. But the Candida albicans were resistant to any percentage of the extract concentration as the result of the zero zone inhibition. And the comparison for both table is both aqueous and ethanolic garlic extract table will show that the highest concentration, which is 100% of the extract, has a larger diameter of zone inhibition comparing to other extract concentrations. Discussion below will support my study based on the result that I obtained. Okay. Mozaffari et al. Discovered the alcoholic garlic extract was considerably more stable than aqueous garlic extract. Next is the Pundir. Pundir highlights that the garlic ethanolic extract inhibited the development of all food-associated bacteria. The bacteria involved were Bacillus subtilis, E. coli, and Staphylococcus aureus. Next is the Caripia and Rajaram discovered that Allium sativum ethanolic extract had antimicrobial activities toward the various tested antibiotic-resistant MAR bacteria, including the Pseudomonas aeruginosa, E. coli, Bacillus species, Porter species, Enterobacter species, and Staphylococcus aureus. Fifth, conclusion and recommendation. The conclusion of the study proved that the garlic has antibacterial potencies against Staphylococcus aureus and E. coli, which only shows the higher extract concentration result in the larger diameter of the zone inhibition. Moreover, garlic does not have antifungal potencies against Candida albicans based on the result obtained. Okay, the recommendation for this study, the first one is use a different concentration of the garlic extract, such as use the highest and the lowest concentration to see more significant results, and the various results can be obtained from it. And the second one is use a different type of the bacteria and fungi, such as Aspergillus species and Bacillus subtilis. And the third one is proceed to MIC and MBC, proceed to MIC, MBC, and MFC to further the study. And the last one, use different type of the extraction, such as oil garlic extract, which can be enhance the allicin compound. Okay, the last one is the reference. This is my reference for my study. Okay, thank you for listening my presentation. I would be happy to answer all the questions from the examiners. Okay, Doctor.
Nurdina.
Yes, are you, Doctor?
You are doing scientific research, right? Lab work.
Yeah.
Okay.
Okay.
All right. Can you show me the tables? The table just now.
Table. All right. Wait. Okay. This is my table. Okay.
All right. So so if I see here, right?
Yeah.
What can you explain between the positive control and the
negative control?
Yeah, positive control and the results that you obtain here.
The positive control that I obtained is a susceptibility antibiotic. Wait, let me re-answer back. Okay, the positive control that we use, that I use in this study, for Staphylococcus aureus and E. coli and Candida were different because of the percentage, the susceptibility of the garlic and the bacteria itself.
What I mean is when you compare your results, right, with the positive control, right? So how many times or how many folds, the mean of diameter zone inhibition, of the garlic, for example, here is the aqueous extract, if compared to the positive control. How many times?
How many times? 3. I did for a 3 time.
No, that one is the replications, right?
Yeah.
But my question is, right, if you compare your results, right?
Yeah.
With the positive control. Right. Okay. So how good is your results? The one that you use is different concentration of the aqueous extract, right? For 100, and then for 75, and then 50. So if you compare with the positive control, right?
Yeah. Okay.
So what can you explain from these results?
Based on the result that I've obtained, the different result from the positive control and the concentration of extract were a lot of different, which is
which is better? The positive control or your results?
I could say that the positive control is much better than the different concentration of extract, because maybe because there's some aseptic technique problem that could lead to the result of my finding, so that the least diameter of zone inhibition that I could obtain in this study. So I would like to answer the positive control is much better to see the diameter zone inhibition.
The positive control is much better because of the technique, or how?
Maybe because of the concentration of the antibiotic itself.
All right. Okay.
Time's up, not enough. So thank you very much.
Sorry, doctor, for misunderstanding the patient.
Okay. Okay, so I would like to call Amira Shazliana for the second presenter.
Okay, I will present my slide now. Can you see my slide?
No.
Not yet.
Okay.
Okay.
Done? Okay, wait. Okay. Okay, wait. Okay, I will start now.
Yes.
Assalamualaikum and very good morning to our panel lecturers and my fellow friends. My name is Amira Shadlana Mitrosidi and I will be presenting my review report on diversity of bacterial endoglucanases. For the introduction, cellulase is known as a diverse group of enzymes generated by fungi and bacteria, including actinomycetes. It also has served a crucial part in the natural decomposition cycle, where plant cellulosic biomass matters are effectively broken down by cellulotic bacteria. Furthermore, endoglucanases are also the enzymes that will help in the conversion of plant material to sugar, where it will cause the breakdown of internal glycosidic bonds in cellulose. The combination of those three major forms of enzymes, endoglucanase, exoglucanase, and beta-glucosidase, are responsible for a complete cellulose hydrolysis. The problem statement: findings have reported a range of factors that contribute to thermostability. Apart from that, it was suggested that thermophilic bacteria that survive in extreme heat temperature can develop thermostable endoglucanase. The objective of this study is that it aims to demonstrate the effect and diversity of bacterial endoglucanase as well as the industrial applications of the enzyme. The significance of this study is that endoglucanases are thermostable that can withstand extreme pH changes and carry out their optimal activity at higher temperatures. The research review, which involved two main bodies. The first one is the structure and the catalytic function of cellulase, and the second one is the thermostability. In the first one, I will talk about the two types of cellulase system, where the second one, the thermostability of the enzyme, I will talk about the thermostable enzymes that can endure extreme temperatures. The first one, structure and catalytic function of cellulase. Cellulose systems are classified into two types, which are non-complex and complex. Aerobic cellulotic microbes will create a non-complex cellulose system, which is a collection of extracellular cooperative enzymes. Meanwhile, anaerobic microbes will produce a complex cellulose system, which is also known as cellulosome. Apart from that, endoglucanase will split beta-1,4-glycosidic bonds at random in the interior of cellulose chain. Exoglucanase will hydrolyze the ends of oligosaccharide chains to release cellulose, which cellulose is then hydrolyzed to glucose by beta-glucosidase. Based on the figure, it shows a representation of cellulose hydrolysis. The endoglucanase will break down the cellulose chains by targeting internal site, hence will produce new chain ends. And then the exoglucanase will release cellulose or glucose by removing the subunits at both reducing end and non-reducing ends of the cellulose chain. And also, it was reported that endoglucanase was discovered to have a weaker attack on the non-crystalline area compared to exoglucanase. Moving on to the thermostability of the endoglucanase. Thermostable enzymes are extremely stable at high temperatures, whereas mesostable enzymes denature, hence lose their optimal activity. Furthermore, thermostable cellulases will enable the utilization of higher substrate concentration when the viscosity of the substrate declines. As a result, this will enhance product yields while lowering capital and processing costs. Not only a low thermostability, but the loss of catalytic activity of the endoglucanase at industrially needed temperatures will restrict their commercial uses. Thermostable enzymes also have a significant advantage where they act as catalysts in the industrial processes, as high temperatures commonly obtain greater enzyme penetration. Research gaps or question in the study is that the first one is important to address that better tools are needed which to characterize and compare enzyme reactions. Next, the protein levels of bacterial endoglucanases are underexplored and not fully studied. And then, what are the genetic sequences of the bacterial endoglucanase? Furthermore, there is no clarity on the DNA technology approaches on the endoglucanases activity. Apart from that, what is the kinetic and the thermodynamics constraints of endoglucanase that act synergistically to break down the polymer? Next, we're moving on to the analysis and the interpretation, where it involves the industrial applications of endoglucanase. Endoglucanase polypeptides have demonstrated an improved performance in the textile industry, specifically in biostoning and biofinishing. For example, trichodermaceae acidic endoglucanase 2 has been identified as a highly effective choice for biostoning. Next, endoglucanases are also extensively used in the pulp and paper industry in order to manufacture cellulose nanocrystals and fermentable sugars. On top of that, endoglucanase could also execute saccharification as well as fermentation at the same time, as it was acidic, thermostable, and resistant to ethanol. Consequently, it becomes advantageous to the industry's manufacturing of bioethanol. The example for the pulp and paper industry is that most cotton garments turn fluffy and drab due to the presence of partially detached microfibers on the surface. Therefore, the use of cellulases can eliminate these microfibers, thus provide a smooth surface as well as the natural color of the clothes. In conclusion, endoglucanases have been extensively used in the breakdown of cellulosic biomass. However, despite numerous efforts, utilization of these enzymes is still typically limited by the cost of the enzymes. Therefore, the scientists must use such an algorithm and machine learning-based technologies on industrially valuable strains and enzymes to increase optimal applications. Continuous research is also important to meet and satisfy the increasing demand for cellulases, as well as to maximize their potential in biotechnology and research. These are the references that being used in our review article. And that's all from me. Thank you, and I will allow some questions from the panels.
Okay, Amira.
Yes.
You are talking about endoglucanase, isn't it?
Yes.
You're studying on endoglucanase enzyme to break the glycosidic bonds in cellulose. Am I right?
Yes.
So is this cellulose branch, highly branch, or what in terms of their punya structure?
The structure.
The structure for cellulose.
The cellulose system, is it?
The structure of cellulose, is it highly branch or not?
It is the cellulose structure is highly branched because it will break down the cellulose of plant cell walls into simple sugars that can be transformed to other products.
So if slightly branched, this endoglucanase will break the bonds at which glycosidic bonds, at branch or linear?
Linear. I'm sorry.
Linear.
Yes.
So the bonds, is it 1,4 alpha beta 1,4?
Beta 1,4 glycosidic 1.
Beta
1,4
1,4
glycosidic 1.
beta 1,4 glycosidic bonds.
Yes.
Okay, just now you're talking about analysis and interpretation under industrial application.
Yes.
So I can see several in your slide. Yeah, I can see several information there, but unfortunately you didn't put the citation, so we don't know which journal you are referred to. Yeah, in your presentation just now. Can you show? Oh, time's up. Yeah, so it's okay. So actually, for better presentation, you should add your citation because you refer to a few journals.
Okay. Thank you for your
Okay,
comment.
so that's all.
Thank you, Doctor.
Okay, welcome. So our third presenter is Amirul Hakim.
Yes, Doctor. Thank you.
Okay,
Can I present my slides?
sure.
Can I start now?
Sure.
Okay, thank you. Assalamualaikum and a very good morning to our respected panels and my fellow classmates. My name is Amirul Hakim bin Rusdi, and today I will be presenting an article review titled as Bacterial and Fungal Cellulase, How Are They Different? I will be presenting the article review in 6 parts. First and foremost, the introduction. There are a lot of microorganisms that are capable of degrading and exploiting cellulose as energy and carbon sources, including bacteria and fungi. Enzymes like cellulase are synthesized by microorganisms during their growth phase on cellulosic materials, such as fruits and woods. Cellulase is one of the many other enzymes that are known produced by several bacteria and fungi. Cellulase enzyme works by breaking down the cellulose molecule into simple sugars such as glucose, fructose, and galactose. Microorganisms such as bacteria, which has high growth rate compared to any other organisms, has a very good potential to be used in a cellulase enzymes production, and cellulase producing bacteria can be found in decaying fruits and many more. Fungi, on the other hand, plays an essential role in nature's cellulose decomposition, which are responsible for 80% of the cellulose breakdown in forest ecosystem. The aim of this article was to differentiate the bacterial and fungal cellulases based on previous researches and related articles. Moving on to the next part, which is the body of the research. Cellulase is a type of enzyme that is mostly made up from exoglucanase, endoglucanase, and beta-glucosidase. A normal cellulase enzyme structure consists of 2 domains, which are catalytic domain and cellulose binding domain, CBD, then known as a carbohydrate binding module, CBM. Cellulose hydrolysis, which is the process of cellulose decomposition, requires this complex to take place. Cellulases are the enzymes that catalyze the hydrolysis of 1,4-beta-deglucosidic linkages in cellulose, and they serve an important role in nature by recycling this polysaccharide, which is the major component of plant cell wall. Cellulose yields appear to be influenced by a complicated interaction involving a range of factors, such as pH value, medium additives, temperature, inoculum size, the presence of inducers, growth time, and so on. In recent years, culturable bacterial cellulase have been isolated and extracted from various sources. Bacteria, which have a faster growth rate than fungi, have a high potential to be used in the production of cellulases. But bacteria are not widely implemented in the cellulase production scene. Fungal cellulases, however, are well-known enzymes that are being studied widely and be used in a variety of industrial processes. A lot of research on cellulases has been dedicated to fungi since it generates a full set of extracellular cellulases rapidly in significant numbers. Then, the research questions that can be identified in this article review are: what is the aim of this research? How to determine the differences between bacterial cellulase and fungi cellulase? How different type of microorganisms produce different type of cellulases? What are the examples of bacterial and fungal cellulases? And what are the conclusion of this research? Moving on to the analysis section, cellulases are comprised of exoglucanase, endoglucanase, beta-glucosidase, and cellulases are the main enzyme for the conversion of cellulose into simple sugars. The visual below shows the mechanism of cellulysis. Fungi are active cellulose decomposers. Fungi are the main agents of decomposition of lignin and cellulose. Fungal cellulases are structurally simple, modular enzymes with two commonly domains, which are carbohydrate binding domains and catalytic domains. These two domains are connected either a thiorin-rich or a serine-rich polylinker, which varies in structure and length. The modules are full of extrapolar amino acid that can stop the substrate moving during the catalysis. The active location of the catalytic field might be topologically shaped tunnel or pocket shape to effectively hydrolyze the substrate. Industrial sectors prefer fungal cellulases since it is extracellular, adaptable in nature, and typically released in high amounts during growth. This is the visual example of Trichoderma rhizae, which is one of the most widely examined fungi cellulase. According to the IUBMB enzyme nomenclature, bacterial cellulases are classified into the group EC 3.2.1.4. Bacteria can grow on cellulose and produce enzymes that catalyze the degradation of cellulose-soluble derivatives or the amorphous regions of cellulose, but only few can synthesize the complete enzyme systems that can affect extensive hydrolysis of the crystalline material found in nature. These few bacteria are called true cellulitic bacteria, and those bacteria that produce some endoglucanases and beta-glucosidases, but not the complete system, are called pseudocellulitic. There are various reasons for bacterial cellulases be used instead of other microorganisms, which are frequent increase of bacterial growth in the multi-enzyme complexes that provide increased functions and activities, thus allowing for more production of cellulose enzymes. Attached here is the visual example of Ruminococcus albus, which is one of many true cellulitic bacteria. In conclusion, both bacterial and fungal cellulase are crucial in today's biotechnological field. Both have its own importance and various use in industrial sector, such as production of detergent, food, and paper. Thus, the cellulase production sector has its effects on significant growth of local economy. It is suggested for the future young researchers to do a thorough research on what type of cellulases that is more suitable for their experiments. This is important due to the fact that each cellulose produced by different sources varies. Thus, it will have a significant impact on the experiment. Last but not least, here are the references that have been referred during the article review writing. And I think that's all from me. Thank you so much for your time.
Okay, Amiru.
Yes.
The same questions as what I asked Amira just now about cellulose structure.
Okay.
So what do you think? Is cellulose highly branch, or linear structure?
In my research, in my knowledge, I think cellulose is an example of unbranched polysaccharide.
Okay, thank you.
Okay.
Assalamualaikum Amira.
Okay.
Sorry.
Waalaikumsalam, Media.
It's okay. Azrina can proceed.
Okay. Typically a review will cover the last 5 years. I nampak banyak 1976, 1985. Lama tu The rule of thumb
Yeah.
is to go back at most 5 to 6 years. Roughly 80% of all cited work should be less than 5 years old lah.
So I should find more related articles on research or recently few years back.
5 to 6 years lah.
Okay.
Thank you.
Thank you.
Okay, ada masa lagi ke boleh tanya? Okay, can you go back to the your title, Miro?
Oh, yep.
Title. Okay, so here this question is how are they different, right? So, okay, you do the comparison between the bacteria and fungal cellulase, right? So, what criteria that you see it different? Can you tell us in points what are the criteria? Based on what criteria?
In my research, based on my title, I do a general criteria in differentiate these both type of cellulase producing
type of
microorganism.
cellulase. Okay,
Yep.
next.
But in my research, I found out that fungal cellulase produce extracellular cellulase. It means that they produce the cellulase to be used outside of the cell. But in bacteria, mostly bacterial cellulase produce cellulitic enzymes in a complex associated with the cell wall, which is called cellulosome.
Okay, so the difference is on type, and then what else? I ask you time. The criteria is not the ni. Okay, so that's all. Okay, thank you, Amirul.
Sorry, because time's up. Okay.
Thank you for all the questions. Thank you so much.
Okay, you're welcome. Now I would like to invite the fourth presenter, Ms. Rena Irina.
Can you see my slide?
Yes.
Can I start?
Yes, please.
Assalamualaikum to all panels and friends. My name is Nurina Adriana binti Noor Afendi from AS2016A3. My presentation today is about a review on in vitro regeneration of economically important plant species. To firstly introduce my topic, in vitro regeneration is a process in which explants of desirable parent plant form organs and tissues after undergoing cell division in sterile condition. Factors influencing in vitro regeneration includes the type of explant, type of culture media, plant growth regulators, sterilization of explants, and culture condition. Economically important plant species are plants that are useful either directly, such as for food, or they can provide raw materials that can be an important source for the economy. There are 5 research questions for this review, which are: what is the aim of this review article? Do auxins and cytokinins induce root and shoot formation? Do plant growth regulators give the same effect to different plant species? Do different concentrations of plant growth regulators produce different outcome? And lastly, what is the gap of the study? The aim of this review is to determine the most effective plant growth regulators for in vitro regeneration of rice, wheat, and bamboo. Out of all the factors influencing in vitro regeneration that I have mentioned previously, my review mainly focuses on plant growth regulators. Plant growth regulators are chemicals that we use to regulate plant growth. There are 5 groups, which are auxin, gibberellin, cytokinin, ethylene, and abscisic acid. Auxin induce root formation and cytokinin induce shoot formation. Auxin and cytokinins are by far the most important for regulating growth in plant tissue culture. For the first species, which is rice, Binte Mostafis et al. 2018 studied the in vitro regeneration of rice using 4 different rice cultivars. They used seed explants cultured on MS medium supplemented with BAP, kinetin, and NAA, and they achieved a regeneration rate of 82% on cultivar MR220CL2, and they achieved 92% of acclimatization. Wagiran et al. 2016 used mature seed explants of Malaysian upland rice seed cultured on MS medium supplemented with BAP, kinetin, NAA, and TDZ, and they achieved a maximum regeneration rate of 100%. Rooting was done on hormone-free MS medium, and the picture shows the acclimatization of Malaysian upland rice seed. Next, Bahorupi et al. 2015 studied 4 different rice cultivars, and they used mature embryos cultured on MS medium supplemented with BAP, NAA, proline, and glutamine. They achieved the highest regeneration rate of 83.2% on Pawana cultivar. Rooting was done on the same medium, and the picture shows the acclimatization of Pawana cultivar. For the next species, which is wet, Asif et al. 2016 studied in vitro regeneration of 4 different cultivars. They used seed explants cultured on MS medium supplemented with BAP and kinetin, and they achieved a regeneration rate of 90% on both AAS11 and PAK13. Next, Zulaj 2019 used embryo explant of Triticum aestivum L cultured on MS medium supplemented with BAP and 2,4-D. They achieved 2.5 shoot per explant with 3.4 cm shoot length. After being successfully transferred into rooting medium, they found that NAA grows the highest mean numbers of roots, while IBA produced the highest mean value of root length at 8.2 cm. Next, Valencia et al. 2020 used a computational approach, namely GRNNGA, which is a method that we can use to reduce trial and error to find the best regenerating system for in vitro regeneration of wet. And they found that the best explant is immature embryo, and 2,4-D and zeatin are the best auxin and cytokinin for regeneration. For bamboo, Goyal and Sen 2016 used mature embryo explant of Dendrocalamus strictus cultured on MS medium, supplemented with BAP and 5% coconut milk, and they achieved regeneration of 10 shoots per flask. 80% of the shoots then develop root when cultured in medium supplemented with IBA. They achieve 70% of acclimatization using a soil mixture of equal parts of perlite soil and farmyard manure. Venkatachalam et al. 2019 used nodal explant of Bambusa arundinacea cultured on MS medium supplemented with BAP and IBA, and they achieved 87.2% of shoot bud initiation. When cultured on medium supplemented with BAP mixed with coconut water and sucrose, they achieved 95.2% of shoot multiplication. 85% of rooting was developed on medium supplemented with IBA and silver nitrate. 92% of survival rate was achieved for acclimatization using a ratio of 3:1 of soil and sand. In conclusion, the best medium formulation for in vitro regeneration of rice is by using mature seed explants with MS medium supplemented with BAP, kinetin, NAA, and TDZ, producing a maximum of 100% regeneration rate. For wet, 2,4-D and zeatin are the best auxin and cytokinin for shoot regeneration. For bamboo, the most responsive plant growth regulators are BAP and IBA mixed with coconut water for shoot regeneration. For root regeneration, IBA and silver nitrate works the best. For acclimatization of bamboo, a mixture soil with a ratio of 3:1 of soil and sand were used to produce 92% survival rate ex vitro. In general, the best type and concentration of plant growth regulators differ depending on the type of explant and plant species. The recommendation for this review is to use activated charcoal to improve cell growth and development and to stimulate nutrient uptake for in vitro regeneration of these economically important plant species. These are my reference. That is all from me. Thank you.
Okay, panel. Q&A session.
All right. Dr. Lina, want to ask question or I?
You can proceed, Doctor.
Okay. Can I see your topics, Nurina? Okay. This is the in vitro regeneration, right? Okay. If you look at the in vitro regeneration just now, right? You mentioned about the shoots, right? The shoots and also the roots, right?
Yes, doctor.
Okay. So the percentage that you showed just now is the percentage of regeneration, right? The percentage of the, okay, we look at the percentage. Okay, if you look here, the these are the regeneration. Where is the regeneration? Okay, some of the review is on the shoot bud initiation, right?
Yes.
Routing. How about the in vitro regeneration? Shoot regeneration? There is there any shoot regeneration? So 95. Okay, for bamboo. How the bamboo just now you concluded which one is the best one is the Bambusa. The which one? Bambusa arundine, right?
Yeah.
So which one is the best one? Bambusa arundine.
Yes.
Okay, so you compared with another one is Dendrocalamus strictus, right?
Yes, doctor.
So it is different species, right?
Yes.
And it is also different. Okay, for Bambusa, how many types of how many review that you compare?
3.
Only 3.
For each journal. For each species, I use 3 journal.
Oh, you use 3 journals, right? And then only 3 journals and on different species.
Yes, doctor.
And then you have different plant growth regulators.
Yes, doctor.
Okay, so meaning that you review it in different species, right?
Yes, doctor.
Okay, why you choose this species?
Because bamboo is one of the.
No, I mean why you choose Dendrocalamus? Why you choose Bambusa arundinacea?
I choose species that the journal shows result of acclimatization.
show result of acclimatization. So other doesn't show result of acclimatization.
Yes.
Time's up. So sorry.
Okay.
Thank you, Doctor.
All right.
Okay, so our next presenter here, the first presenter is Anis Nazifa.
Hello, can you hear me?
Yes.
Can hear but
Okay.
slow.
Okay.
Okay,
I'll,
louder. Okay, now
I'll
can hear.
present my slides. Okay. Can you see the slides?
Yes.
Hello? Oh, okay. Okay. I will start my presentation. Assalamualaikum and a very good morning to fellow panels and students. My name is Anis Nazifa binti Anaruddin and I'll present my final year project. A composite is typically characterized as the combination of two or more unique materials where one serves as a reinforcement while the other as a polymer matrix material. Synthetic polymers have great qualities such as stability and permeability. However, its usage poses threats to the environment. Biocomposites are essentially the same, but the materials are derived from biological origin, for example, using natural fibers for reinforcement, as it have advantages such as cheap and lightweight. The interest to use natural fibers as reinforcement in polymer composite grew as it can replace the usage of synthetic polymers. Bamboo fiber have improved mechanical qualities than most due to the additional lignin content, as seen here, as well as the least amount of moisture absorption. The research questions of my studies are how does bamboo fiber combine with polymer matrix, how is the bamboo fiber being extracted, and how is bamboo fiber reinforced composite being manufactured? Can the performance of BFRC be improved by alkalization treatment, and how does bamboo fiber improve the properties of BFRC in automotive applications? The objectives of this study is to provide a review on current developments and research on bamboo polymer composite, as well as to study the applications of bamboo polymer composite in the automotive industry. The content of the studies are bamboo and fiber extraction techniques, BFRC manufacturing techniques, BFRC for automotive applications, modification process to achieve greater performance, and bamboo hybrid composite. Bamboo fibers are easy to cultivate and grows abundantly in nature. It has a comb resembling an empty cylinder with a diaphragm that displays a ring-like configuration, and this ring splits the core of each comb. The gap within two rings where the branches develop is referred to as internode, and bamboo fiber accounts for over 90% of bamboo's total weight. There are two kinds of extraction involved in acquiring bamboo fiber, notably mechanical and chemical process, both of which start with the splitting of bamboo into strips. The chemical method entails the production of cellulose fiber by alkali hydrolysis, followed by multi-phase bleaching through carbon disulfide. Mechanical process use enzymes to treat the cut bamboo, resulting in development of spongy mass from which single fibers are retrieved using mechanical comb. This figure here shows the steps for extracting bamboo fiber, and depending on the manufacturing process, it is useful to separate into rough or fine bamboo. Bamboo fiber can be mechanically extracted using various methods, and this figure here shows the most frequent types of extracted bamboo, which are strips, flake, long, long fiber, short fiber, sliver, and powder. BFRC have been manufactured using a variety of process, including hand layup, operation molding, and melt blending. Hand layup method is one of the most basic and common polymer manufacturing process, and this technique is best used for thermosetting polymer-based composite as it requires less cost and equipment. The fiber are placed into a mold layer by layer, alternating with resin, and let cure for 24 hours at room temperature to allow cross-linking of fibers and matrix. The mechanical properties of BFRC are determined by the modulus and strength of fiber, chemical stability, stiffness and strength of matrix, and interfacial bonding strength between bamboo fiber and its matrix. Fiber length, volume fraction, aspect ratio, fiber matrix addition, and fiber positioning also affects the mechanical properties of BFRC. For instance, fiber length determines the composite's tensile modulus. Long fiber has higher tensile modulus than short fiber as it may withstand higher weights attributed to the rise in transfer length. For fiber volume, excessive fiber in composite materials can degrade mechanical characteristics caused by lack of adequate fiber matrix bonding, which disrupts transmission of fluid to the connecting fibers. Biological factors that can affect mechanical properties of BFRC includes weathering and soil burial. This is caused by moisture absorption from bamboo fiber. Bamboo fiber is hydrophilic, thus absorbs water and swells, leading to formation of micro cracks in the matrix and weakens fiber matrix interface. Automotive components such as seats, floor mats, dashboards, door panels have used BFRC as it offers high stiffness and strong thermal and acoustic insulation. In comparison to other panels, door panels using BFRC provide the lowest weight and stress value due to high stiffness and lightweight of material, and door panel using BFRC offers the advantages of lower cost, increasing customer experience, and lower fuel consumption. Surface modification treatment can considerably enhance the characteristic of fiber and composite. Chemical treatments have been discovered to increase the adhesion, which is one of the key parameters determining the mechanical characteristic of composite. Most common technique is alkali treatment, which involves the immersion of fibers in concentrated sodium hydroxide solution. The NaOH interacts with hydroxy group of fibers to eliminate the substance that covers in a higher surface roughness and fiber matrix addition. The mechanical
Dah lost tak dengar.
Internet problem.
Ya, kita tunggu dululah ya.
Ya.
Contact dia melalui grup kot.
is enough.
Okay, dah ada.
Anis, do you have problem with your internet? We can't hear you actually.
Oh, I'm sorry. From which slide?
Go to the previous one. Yes. Go to before suggestion and recommendation. Before this slide.
Oh, dang.
Okay, move to the previous slide. Hello.
Okay.
Move to the previous slide before
I'm
suggestion.
sorry. I before suggestion. Okay, here. I lost connection earlier.
Sebelum lagi, satu lagi.
This?
I think sebelum lagi. Kat sini.
Okay. I apologize. I lost connection and I didn't realize.
Okay, let me start again. Surface modification treatment can considerably enhance the characteristic of fiber and composite. Chemical treatments have been discovered to increase fiber matrix addition, which is one of the key parameters determining mechanical characteristic of composite. Most common technique is alkali treatment or Mosturizer, which involves the immersion of fibers in concentrated sodium hydroxide solution. It eliminates substance covering the external surface of fiber, including hemicellulose, lignin, wax, and oils, resulting in a higher surface roughness of fiber matrix addition. The mechanical characteristics of composite, particularly tensile, flexural, and impact strength, are affected by whether the fibers are treated or not. However, it is observed that treatment with a high concentration of NaOH can eliminate cellulose and damage the structure of fibers, leading to a reduction in fiber strength. 5% of NaOH solution treatment is enough to alter the surface without compromising fiber strength or mechanical performance. Nowadays, the use of two natural fibers to create a hybrid composite is useful for vehicle manufacturers in an attempt to substitute synthetic fiber and achieving better composite characteristics. Hybrid composites are cost-effective, eco-friendly, and has considerable mechanical strength alongside low density. There is a lot of potential for BFRC to be used widely in transport. However, further research of BFRC is needed to uncover the latent potential of innovative natural fiber composites, especially as polymer composites have received a lot of interest in the automotive industry in recent years. In conclusion, natural fiber-based composites are excellent for future use in the automobile industry due to their biodegradability, renewability, and non-toxicity. Bamboo fiber stands out due to their great environmental flexibility and strong tensile strength. And bamboo fiber reinforced composites are durable, dimensionally stable, and may be designed to any shape at a cheaper cost than traditional ones. That is all for my. No. This is the reference I use for my presentation, and that is all.
Assalamualaikum Anis. Assalamualaikum Anis.
Waalaikumsalam. Yes, saya. Yes, Doctor. Yes, me.
From your point of view, what is the most effective methods in extracting bamboo fibers? Yang you dah sebut tadi.
In my opinion, the best way to extract bamboo fiber is by using mechanical techniques. Although the procedure, the technique is less cost-effective than chemical approach, it is more environmentally friendly. And as the main reason we use bamboo fiber reinforced composite is for its renewability and sustainability. So it would be better to choose the method to extract it that is more environmentally friendly than not.
Okay, thank you.
Okay, in the last slides, you have mentioned about the bamboo can be renewable. What does it mean?
Bamboo are renewable due to its large abundance and easily cultivated in comparison to other natural fibers. And bamboo fiber is found abundant all around the world in South Asia and Africa as well. So it is easily obtainable.
All right. Okay.
Okay, time's up.
Okay. Thank you.
Okay, so our next presenter, Nur Izzati.
Yeah, Assalamualaikum.
Okay, Waalaikumsalam.
Okay, I will share my slide. Can you see my slides?
Yes.
Okay, I will start my presentation now.
Ayat, please.
Assalamualaikum and a very good morning to all panels and my fellow friends. I am Nur Izzati binti Rahim, and today I am going to present my final year project with the title of the Development and User Acceptance Survey of Astaxanthin Infused Topical Moisturizer. Microalgae are organisms that have a wide range of biotechnological applications, such as nutraceutical applications. Hematococcus pluralis is one of the microalgae that produce a high amount of astaxanthin that gives many benefits to human health, including skin benefits. Astaxanthin is used in skincare products such as moisturizer because of its strong antioxidant. Despite its endless benefits to human skin, the application of astaxanthin in topical moisturizer is still limited because of the data of incorporation of astaxanthin in topical moisturizer is still low. Thus, this study will be an additional reference for the future research of incorporation of astaxanthin in topical moisturizer. This study also will contribute to further information regarding consumers' response to the newly developed moisturizer based on the user acceptance survey. The objectives of the study is to optimize the ratio of astaxanthin, honey, and Neutrogena gel moisturizer via box Benken design. The second objective is to conduct user acceptance survey of the astaxanthin moisturizer comprising of appearance, odor, and also texture on 20 respondents. Astaxanthin is a reddish pigment that belongs to a chemical group called carotenoid. Based on the journal by Seki et al. 2001, in clinical studies, it is proven that astaxanthin can increase skin moisture, retention, elasticity, and increase skin smoothness. Box Benken design was used in this study. Box Benken design is the response surface methodology design that require three different levels, which is -1, 0, and also +1. Box Benken design can assist in creating the new formulation by conducting the response surface analysis. So this is the flowchart of the methodology. The first step was the extracted astaxanthin in Malaysian virgin coconut oil from H. pluvialis was obtained from the microalgae research laboratory. The second step was the preparation of astaxanthin moisturizer. This astaxanthin moisturizer were developed by mixing three ingredients, which is Neutrogena gel moisturizer, astaxanthin, and also honey. 15 samples were developed with different ratio and formulations by using box Benken code. The third step was user acceptance survey. In this survey, three attributes which consist of appearance, odor, and texture were being graded by the respondents. The last step was statistical analysis. And this is the preparation of the astaxanthin moisturizer. As you can see in the table, the three ingredients were being supplied at three different levels, which is -1, which is the lowest level, and +1 is the highest level. Moving on to the results and discussion, as you can see in the table, this is the raw data that I collected from the user acceptance survey. This is the total score from the 20 respondents. Next, I will explain about the response surface plots. Based on the raw data that I get, I'm doing the analysis and this is the result that I get. In terms of appearance, the interactions between astaxanthin and honey, astaxanthin and Neutrogena, and honey and Neutrogena indicates that the respondents likes when the moisturizer have the when the all ingredients are at the higher concentration, which is at the level of +1. In terms of odor, the respondents likes when the interactions of astaxanthin and honey and astaxanthin with Neutrogena shows that the respondents well accepted when the ingredients is at the higher concentration, which is +1, while the interactions between honey and Neutrogena shows that the respondents like when honey at the lowest concentration, which is at level -1, and Neutrogena at the highest concentration, which is +1. Okay. In terms of texture, the interactions between astaxanthin with honey, astaxanthin with Neutrogena, and honey with Neutrogena shows that the respondents likes when all of the ingredients at the higher concentrations. Okay. Moving on to the response surface optimization plot, this is the simulated data. Okay. Based on the data, the composite desirability is 69%. The 69% indicates that only 69% of the 20 respondents likes the moisturizer when the ingredients of Neutrogena gel moisturizer is at 20 ml, astaxanthin at 1 ml, and honey at 0.9192 gram. Because of this pandemic, insufficient data was collected. Thus, it leads to the a little bit low of the composite desirability. In conclusion, the astaxanthin moisturizer was successfully developed by using box Benken design. The best formulation was found to be when Neutrogena gel moisturizer at 20 ml, astaxanthin is at 1 ml, and honey at 0.9192 gram. It is recommended to increase the number of respondents and also expand the demographic. The respondents should be from various places, age, and also race. This is the reference that I use. That's all from me. Thank you.
Okay, thank you, Izati. This is your research, right? You carry out this research?
Yes. Yes.
Where did you do your research? In FSG lab?
I'm doing it, no, at home only.
At home only. Okay.
Yes.
Okay, you add honey, 0.9192 gram. May I know what type of honey? Is it beef honey, madu lepah, madu kelulut, atau madu macam mana?
Manuka honey, raw Manuka honey.
Raw Manuka honey.
Because Manuka honey is suitable to use for our skin.
I see.
Yeah.
So maknanya you belilah.
Yeah, memang.
Okay.
My father use it at home, so I just use it.
Okay, so your respondent age, may I know, because 20 respondents, is it the same age or various age?
No, various age, from 20 years old and above.
20 and above,
Yeah.
Because maybe different levels, different age actually different preferred skin moisturizer, maybe,
Yeah.
Okay, may I see your table once again? Result in the tables just now.
This one?
Yes. So what does it mean? Appearance 100%, order 80%, texture 90%, 90 not 90%. What does it mean score?
This is the total score.
The total score
From
meaning?
the 20 respondents.
Okay.
As you can see, the lowest score is 7 and the maximum score is 9. So I just total up all the score and this is the value that I get.
Okay. So go back to next slide.
This one?
So based on this one, which one is the preferred? Nampak sama kan, tapi ada tak yang lebih dia mereka gemari? Astaxanthin with honey, astaxanthin with Neutrogena, or honey with Neutrogena?
It is actually, the same actually. Just different graph to see more detail. But
So you cannot determine the most preferred, right?
Okay,
Sebab sama
yeah,
saja.
yeah.
Okay, how long actually your experiment you did in at home?
Like 6 to 7 weeks.
6 to 7 weeks
Yeah.
only. Okay, so that's all. Thank you.
Okay, thank you so much.
You're welcome. So next presenter Nurliana Hazwani.
Kak Azani, boleh tak kita ambil gambar dulu?
Boleh. Oh, sebab ini the second one, isn't it? Betul.
So yang the first 6 tadi tu kalau nak keluar dah boleh dah lah. So from Nurliana Hazwani, Anissa, Indah, Izzah, Siti Hajar and Ros Nuzwir. Okay, sekejap.
Another 6 students,
Okay, sebab saya nak ambil gambar, boleh tak tolong buka kamera masing-masing sekejap?
Nuriza lagi sekali.
Ni tak semua ada ni. Sekejap, Liana Hazwani ada? Ada. Anissa Ahmad.
Tak semua lagi ya.
Kena semua masuk ni Kak.
Okay, nanti kat dalam grup.
Kakak tolong mesej ya.
Okay.
Indah ada. Indah ada. Nurul Izzah tak ada lagi. Siti Hajar Salim tak ada. Terus Munswi tak ada. Kita tunggu sekejap ya.
Dr. Lina nak saya jaga masa ke?
Boleh juga sebab I pukul 12.30 dah nak kena keluar.
Okay,
ada meeting lain.
Saya jaga masa ya.
Okay.
Thank you.
Alright. Berapa ya? Yang tengok sikit.
Belum lagi.
Ramai ni.
Kan? Tak masuk lagi.
Indah, Yana.
Belum masuk lagi.
Maklumkan dalam WhatsApp group kan.
Azrina dekat mana Azrina? Kat mana
Kat
sini?
rumah, Doktor.
Oh, Shah Alam. Okay.
Doktor kat rumah ke?
Dia kat rumah. Tadi pada Indah dah.
Anissa tolong buka kamera. Siti Hajar buka kamera.
Dah pindah lama.
Ros Nuzul tak masuk lagi.
Seorang lagi.
1, 2, 3, 4, 5.
Seorang lagi. The last one. Ros Nuzul.
Okay. Alright.
Terus siapa ada kontak nama Ros, please call, nak masuk.
Ada. Indah juga. Ada 2 nama.
Indah Nilam ada 2 nama. Okay.
I think for Ros
Rose cakap dia punya Google tak respon, dia suruh tunggu kejap. Internet dia ada problem sikit.
Saya suruh orang buka kamera, saya tak buka kamera. Aduh.
Okay, Ros dah join ya.
Okay, Cik Ros, okay. Alright, senyum semua. 1. Siti cepat. Okay, 1, 2, 3. Sekejap, jangan lari. Okay, satu lagi. 1, 2, 3. Okay, that's all. Thank you.
Terima kasih Lina. Okay, so shall we proceed with the students, the next batch of students, which is Nolia Hazwani. Okay. Nolia Hazwani. Student tak dengar ke, tak respon?
Sekejap. Kita tunggu.
Okay dah.
Okay, dah nampak.
Dah boleh start ke?
Nolia,
Liana suara jauh.
Jelas tak?
Sayup kedengaran.
kuatkan suara.
Kejap ya.
Masih perlahan. Nampak sikit.
Dekat mic. Dekatkan dekat mic.
Boleh dengar ke?
Liana, are you using any headphone ke earphone sekarang? Rasanya cuba buka dan cakap biasa je, direct to the top.
Buka. Tadi saya tak buka. Doktor kata slow.
Ni pasang pun tak dengar juga. Jauh sangat.
Sekejap, saya join pakai phone saya lah untuk voice.
Hello.
Kejap. Ada side effect.
Matikan sound dekat laptop.
Dah okey ke?
Okay, better.
Saya mula.
Alright.
Assalamualaikum. My name is Nurliana Hazwani binti Syadani, and today I will present my review article entitled A Review on the Role of Vaccines in Reducing Antimicrobial Resistance. The discovery of naturally occurring antibiotic penicillin in 1928 opened the door to the modern era of medicine. Since then, these magic bullets have saved thousands of lives. However, the misuse and overuse of antibiotics has caused the emergence of antimicrobial resistance, AMR. AMR is a serious public health concern across the world. This is due to the rapid emergence and spread of the resistant pathogens. In recent years, most of the battles against AMR are no longer effective. This is because when a new antibiotic is introduced, the bacteria will quickly evolve and develop resistance against this antibiotic. Therefore, vaccine...
Sorry Liana, could you please open your camera? Sebab kita orang tak boleh nak access you untuk eye contact.
But I have
buka video.
Dia dah buka dah. Dia buka dua.
Okay.
Satu suara, satu kamera.
Alright. Okay, can continue.
Thus, vaccine can play a potential role in reducing AMR. Vaccine can reduce the prevalence of infections that substantially will also reduce the antibiotics prescriptions. Low antibiotic usage will subsequently reduce the emergence of resistant pathogens. Therefore, indirectly, vaccination can diminish and
diminish the spread and circulation of resistant pathogens. This is the research questions of this review. Can vaccine be an alternative tool to decrease AMR? Why vaccine is an effective tool to decrease AMR? How vaccine can reduce AMR? What is the type of vaccine that has potential to combat against AMR? And what can be improved in the development of new vaccine? All of these questions will be answered in this presentation. The objective of this review are to discuss the evidence that vaccine can play an important role in combating AMR and to describe the future prospects of new vaccine development. Now I'm going to describe the types of vaccines. The first one is live attenuated vaccine. It is made from living viruses or bacteria that have been weakened. On the other side, inactivated vaccine use pathogens that have been inactivated or killed using chemicals, radiations, or heat. Subunit vaccine is a vaccine that uses specific parts of the antigens that best stimulate an immune response. Toxins produced by certain bacteria that have been inactivated using chemicals or heat are used in toxic vaccine. As for conjugate vaccine, it is part of the weak outer antigen code of the bacteria or virus that are linked to a stronger carrier protein using chemicals. Among these types of vaccines, conjugate vaccine is under-recognized tool that has been observed to reduce AMR. Thus, I'm going to discuss more on the potential role of this vaccine in reducing AMR. The first role is by restricting the spreading of pathogens through herd immunity. When lots of people in an area are vaccinated, fewer people get infected. Thus, fewer pathogens are around to spread from person to person and therefore mitigate the spread of AMR. For example, the introduction of conjugate vaccines against H. influenza type B hit on infants in Brazil, Canada, Japan, and UK reduced the rate of hip infection and AMR in both vaccinated and unvaccinated children due to the herd immunity effect. The next role is by reducing the use of antibiotics due to fewer infections. By preventing the spreading of pathogens within the populations, vaccine can reduce prescription of antibiotics in treating infectious diseases. This will subsequently reduce the emergence of AMR as a result of less antibiotic selection pressure. This has been proven by the decrease in the total use of antibiotics in vaccinated children in the US after the administration of heptavalent pneumococcal conjugate vaccine, PCV7. The next role is by preventing the misuse of antibiotics. Many cases of febrile disease that may resemble some of the typhoid fever symptoms are usually caused by viruses, which means that the use of antibiotics is unnecessary. However, the release of new typhoid conjugate vaccines, type 4 TCV, have reduced the misuse of antibiotics in treating typhoid fever, thus reducing the selective pressure on bacteria, the growth and spread of AMR. Next, I'm going to explain why vaccine is an effective tool to reduce AMR. Firstly, vaccine resistance is an uncommon occurrence. On the other note, bacteria have acquired resistance to almost every currently available antibiotic. Next, vaccine works prophylactically, which is administered to the individual as a preventative measure before the individuals are infected by the disease. Hence, the probability of pathogens to proliferate and grow in a large population within the body is very low. Whereas antibiotic works therapeutically, which is administered after the symptoms have been diagnosed in patients. Therefore, during the infections, the bacterial cells have grown and proliferated in high numbers. From the previous studies, my suggestion on the future prospect of the development of new vaccine should aim at expanding the coverage of serotype. causing disease. Thus, more effective vaccine with broader coverage will be able to limit this problem. Next suggestion is new target of vaccines. It should target microorganisms associated with increased AMR, a large number of antibiotic use, or hospital-acquired infections, including surgical site infections, as well as gastrointestinal and urinary tract infections. In conclusion, vaccine can be used as an alternative tool to reduce AMR by reducing the spread of resistant pathogens. That's all from me. Thank you.
Okay, Liana Hazwani.
Yes, saya.
Okay, can the vaccine actually lead to the antibiotic resistance?
It can if the serotypes covered by the vaccine are less likely to be the resistant, because if it's less likely to be resistant, then it will increase the selection pressure.
Okay. Can you name one of the vaccine that have to counter the AMR disease?
Typhoid conjugate vaccine.
Pardon?
Typhoid conjugate vaccine.
Okay.
Batifree.
Okay, that's all from me.
Thank you.
Ada soalan lain?
Okay, just now you mentioned about prophylactically, right?
Yes.
So how does the process, or how does it what we call this how does it function?
Prophylactically, it is administered to the individuals as a preventative measure. Means that before that person get infected, we already give the vaccine to the individual, so that if the individual get infected, it will reduce the symptoms. Means that the pathogen also cannot reduce in a large population.
Okay, that's all I guess. Or Dr. Nariza ada soalan lagi? Oh, time's up. Time's up already, ya. Okay, thank you very much. Now we will proceed to next presenter, which is Anissa Ahmad.
Can I start?
Yeah, sure, please.
Assalamualaikum warahmatullahi wabarakatuh, and very good morning to my fellow lecturers. My name is Anissa binti Ahmad, and today I'm going to present on my final year project titled Isolations and Characterizations of PHA-Producing Bacteria from the Municipal Wastewater. The background of the study: polyhydroxyalkanoate PHA is a biodegradable polymer in nature with characteristics that can be used as plastic for a large community. These polymers are produced in the presence of excessive carbon source that accumulate intracellularly in some of microbes if there is a growth-limiting factor such as nitrogen, oxygen, phosphorus, and sulfur. This PHA also become a potential substitution for synthesis plastic due to the various characteristics like crystallinity, elastomeric design, and degradability in a wide range of environments. Plastic has contributed to the environmental degradation by buried daily in landfills and take-out spaces. The processing biodegradable plastic is an alternative way as it can generate a new end-of-life waste disposal opportunities. Besides, the production of biodegradable plastic cannot be fulfilled effectively due to the constraints of material and high production costs. The significance of this study is beneficial to improve the productions of PHA to make it more desirable to be used as an alternative to petroleum-based plastic. This study also contributes to the additions of culture collections and enhance the productions of PHA by exploring free carbon sources and waste carbon. The objective of this study is to assess the PHA-producing bacteria strain from the domestic waste that serve as the carbon source for PHA productions, and to distinguish the morphological and biochemical characteristics of the isolated bacteria. Plastic has become threats to the environment because it's considered hazardous because of its long-term dangers of toxic chemicals emitted in the landfills and groundwater contaminations. The restricted bioavailability and polymer insolubility in water has become the major obstacles in converting the biodegradation of oil-based plastic. PHA-producing bacterial species has taken place in more than 70 bacteria and archaea groups that can be found in gram-positive bacteria and gram-negative bacteria. Gram-negative bacteria contain lipopolysaccharide LPS endotoxin which can co-purify the crude PHA polymer that can cause immunogenic reactions in humans. Thus, the gram-positive bacteria mostly used in biomedical application compared to gram-negative bacteria. The challenge in PHA productions is the lack of evidence on what is the suitable and cheapest way to increase the productions of PHA. For example, based on what type of microbe used, the culture practice, and the carbon source choice, which is obvious that there are other methods can be used to develop PHA. There are 4 phases for the methodology throughout the project, which in the phase 1, the sample were collected from the college mawar treatment plants. The sample were taken for the enrichment of the PHA-producing bacteria. In the phase 2, we are target on isolations of PHA-producing bacteria. In phase 3, to screen for the PHA-producing bacteria, we use sodium black B colony staining, now blue A colony staining, sodium and as well as sodium black B cell staining. In phase 4, for the previous positive for PHA producer were subjected for grain staining, endospore staining, capsule staining, and some biochemical tests. For the colony staining by the sodium black B, out of 50 colonies, only 6 bacterial isolates were tested positive as PHA producer, namely isolate 2, 5, 13, 38, 44, and 47. The darkened color of colonies indicate the positive PHA producer bacteria, while the colonies that appear white are deemed negative because it enables to incorporate the sodium black B stains. It is crucial to screen the bacteria in a short amount of time utilizing PHA-specific stains for granule identifications when extracting PHA-producing bacteria from the nature. The previous positive bacterial isolate were further confirmed by cell staining using sodium black B. Out of 6 bacterial isolates, 3 isolates shows the presence of lipid granules. The 3 bacterial isolates, namely isolate 5, 38, and 44, were confirmed as positive PHA-producing bacteria. The lipid granules can be observed in the form of blood precipitate that are present in the bacterial cell. However, the lipid granule stains were not quite intense. This may be due to the incorrect staining technique. For the morphological characterizations, all 3 of the positive bacteria isolate were gram-negative bacteria. The isolate shows short root shapes and some root shape chain. While for the endospore stains, all the bacteria isolate show the positive for endospore stain where they are producing endospore which can be observed inside and outside of the cell. For the capsule stain, all the bacteria isolate were found negative for capsule. For the biochemical test, all 3 bacterial isolate were tested positive for the catalyst test, which indicate the presence of catalyst enzyme in the bacteria, while all the bacteria were tested negative for the indole, which means that the bacteria cannot break down the tryptophan and thus producing the indole. All the bacterial isolate were found to show the same reactions for triple sugar ion test, TSI, which both deep and slant change color to yellow and shows no productions of gas and hydrogen sulfate. And lastly, for the metal red and Voskert test, all the bacterial isolate were tested metal red positive, while all the bacterial isolate were tested negative for Voskert tests. In conclusions, 6 out of 50 colonies were tested positive for the PHA accumulations bacteria during the first screening using the Sudan black bee colony staining. However, out of 6 bacterial isolate, only 3 are confirmed as PHA-producing bacteria, namely isolate 5, 38, and 44 during the Sudan black bee cell staining. The major limitations that we face to complete the project is the time provided to complete the project, where every step needs to be taken and every step needs to make correctly to minimize the error. And next, the insufficiency of material, where we have to find the material in another labs, and it affected the workflow of the project. That's all for me. Thank you.
Anissa, where do you obtain your samples?
Huh?
Where do you collect your sample?
College Mawar treatment plant.
Okay. So actually, how long does PHA takes to decompose?
Do
you have any idea?
I don't have any idea about that. I'm sorry.
Okay, Anissa. Again, proceed. What proceed tadi now? Never mind.
Continue. You tadi jawab tadi kan?
My question is, what do you mean by insufficient material? You must go to other lab you said just now. Is it chemical or what?
In terms of chemicals, where some materials are not available in the lab in Block B, so I have to go to another labs to another like food test lab to borrow their chemicals.
But you have sufficient material that you need to carry out the test, isn't it? I mean the one that you took from College Mawar, the sample. That's enough for you. Sample enough. Yeah, sample is enough, but the enough is chemical.
Yes.
So how many replicate you carry out the experiment?
Replicate 21 replicates.
21 replicate for each sample, is it? It's quite a lot.
For each samples, I do triplicates. 21 plates in total.
21 for each total, or for each sample?
For each for the total plates. For each sample, I do sterile dilution. For each sterile dilutions, I do triplicates.
Okay. So the rest the question, panels? I guess no more question. So kalau tak ada, we will proceed with the next presenter. So our next presenter is Indah Nilamsari.
Okay, can you hear me?
Yes.
Okay. May I start now?
Yes, you can start now.
Okay. Assalamualaikum and good morning. I bid to all the panels and my fellow friends. Today, I'm Indah Nilamsari, will be presenting my review on the different types of plant growth hormone for Micropropagation of Bambusa vulgaris. This review article is made based on the several reliable experimental researches involving in vitro micropropagation of Bambusa vulgaris. So as an introduction, Bambusa vulgaris belongs to the lineage of Bambusoideae. It is a valuable and versatile species and developing application in many areas such as paper and bioplastic industry, construction, handicraft, and also food. It also contributes in the environment by slowing down the land degradation, aiding environmental conservation, and contribute to 7 out of 17 United Nations Sustainable Development Goals. So, plant tissue culture is the widely used technique for rapid and mass in vitro micropropagation of Bambusa vulgaris, and the plant growth hormone is the chemical substance crucial in the protocol of the technique, and it aids in the growth of the species. So, the selection of suitable plant growth hormone is important to increase the viability, cost effectiveness, and help restore the formation of the species, and consequently extend the application and the commercial value of the species. So, the objective of this review article is to review the plant growth hormone effective for Bambusa vulgaris, which is useful to enhance the existing experimental research and study protocols, apart from improving the quality and quantity of the mass propagation of Bambusa vulgaris. So, the research gap that exists is that the ability of Bambusa vulgaris grow more successfully even with only incorporated hormone without any application of external regulators. And the research questions are: which is the most effective plant growth regulator for Bambusa vulgaris? Do plant growth hormone work best in combination or alone? And which is the best plant growth hormone combination? And lastly, do the type and concentration of plant growth regulator influence the effectiveness toward the growth of Bambusa vulgaris? So, how does plant growth hormone works on Bambusa vulgaris? The response of the same plant regulators are specific to the types and concentration. This is due to the tissue specificity of the cytohormone receptors, and it is achieved via transcriptional activation, initially mediated by the binding to the specific receptor and also signal transduction. So, auxin, cytokinin, ethylene, gibberellins, and abscisic acid has about 10 to 1,000 times more influence toward the plant development as compared to the natural auxins. So, what are the general functions of plant growth regulators to Bambusa vulgaris? They help in the cell differentiation, physiological process, cell elongation, cell purification, coordination, and regulation at the concentration significantly lower than the other vitamins and nutrients that can offer. So, there are also specific functions. For example, auxin, commonly used for rooting formation and shoot multiplication, callus stimulation, or cell purification. For abscisic acid, gibberellins, and ethylene, they are usually used for regulation of somatic embryo development, shoot elongation, and act as an elicitor of the hairy root culture. And lastly, for cytokinin, it is commonly influence the shoot formation, cell division, and also the delay in the tissue necrosis. So the analysis from the review is that the first one, the review on the frequency of certain type of plant growth hormone regulator for in vitro micropropagation of Bambusa vulgaris shows that auxin indole tributyric acid (IBA) is the most commonly used single plant growth hormone. It is because it is used in 6 out of 7 experimental researches that have been made. In terms of the number of root, the maximum result, which is 17.33, was obtained from the micropropagation without implementation of any growth regulator. But for root length, the maximum value is 25.35 cm, also obtained without any implementation of any growth regulator. And lastly, for root percentage, which is the maximum value is 87.12% by 7.5 milligram per liter of IBA. So why is IBA usually used and very effective for Bambusa vulgaris micropropagation? It is because it helps in the root formation in short period of time. It also help in the stimulation of culture root and shoot multiplication, but with greater advantage than other hormones, as it is able to exhibit similar rate of activity as other auxin, but relatively in a lower concentration. It also helps in rooting percentage of the culm and the branch cutting of the Bambusa vulgaris, especially for the plants with lack of endogenous auxin. So another analysis is that the combination of IBA and NAA is ideal for the in vitro rooting of the plant tissue culture of Bambusa vulgaris, and BAP in combination with kinetin show positive result by helping the development of the shoot, particularly in the length and number, while combination of BAP with 2,4-dichlorophenol-oxyacetic acid has intensified the root percentage of Bambusa vulgaris, help in shoot multiplication, purification, and also bud sprouting. So BAP is extremely used or extensively used also because of the autoclave nature and the cost effectiveness. So in conclusion, IBA is proven to be the most effective plant hormone, especially on the rooting development of Bambusa vulgaris. But for combination, the supplementation of BAP in combination with kinetin and NAA is also beneficial for the success of the micropropagation. So as a recommendation, applying IBA as an essential plant growth regulator of Bambusa vulgaris is recommended due to its functionality, particularly in root formation, and also its cost effectiveness. It is also worth to study the other aspect of the plant, which is the type, combination, concentration, and size of plant hormone, also the type and climatic condition of the explant being collected, and lastly, the environmental condition implemented during the in vitro micropropagation of Bambusa vulgaris. So these are the reference from my research, and that's all from me. Thank you.
Okay, Indah, can you go back to your result, table 3?
Okay. Yes.
Okay, this is control. Okay,
Yes, true.
compared to IBA, NAA, and also IAA.
Yes.
0 outside the bracket is mean without plant growth regulator, 0 concentration. Am I right?
Yes.
In the bracket is the number of roots, right?
Yes.
So if you look at the number of roots here, when you compare to IBA, NAA, and IAA, it's a very clear that the control treatments actually produce significantly higher the number of root and also the root length as well, and the root percentage as well. So how comes your conclusion said that IBA just now you mentioned as the best hormone for rooting induction in your conclusion?
It is because it is mainly used in many researches.
Okay.
Researches. It is frequently used.
Mostly used in many research, is it? But in terms of the number of roots, the treatments without IBA much better. So based on this control treatments, what can you make a conclusion for this bamboo based on control treatments? So, you can make a conclusion actually.
Apart from using IBA, it is also advisable to not use any plant growth hormone because it is also effective for the micropropagation of Bambusa vulgaris.
Yes, it's actually control much better than IBA and AIA, even though IBA is actually commonly or widely used by a researcher. But in terms of the number of roots, control treatment much better. How about shoot induction? Any hormones? You do investigation based on your review article?
So the shoot multiplication, it is more in the combination of BAP with kinetin and also 2,4-D.
May I know the concentration? The most suitable concentration?
Concentration is around 2 milligram per liter to 10 milligram per liter.
2 to 10 milligram per liter, BAP alone.
BAP and kinetin. BAP in combination with kinetin or
kinetin.
Yes.
Okay. What is the most commonly used explant for bamboo?
Not nodal segment.
Nodal segments. The age of the nodal segment?
Young age.
Young age, yeah.
Yes.
Okay, what's the difficulties for in vitro regenerations of Bambusa?
The difficulties is in terms of sterilization, and also there are also many problems during acclimatization because the plant cannot adapt with the new ex vitro condition.
Okay, Indaya, that's why because time's up. Thank you for your answers.
Okay.
Okay, next Nurul Izzah.
Nurul Izzah ada?
Azani, own student tak perlu letak markah kan?
Tak perlu evaluate.
Alright.
Student.
Okay.
Can you see the slides?
Yes.
Yes.
Okay, can I start now?
Yes, please.
Okay. Assalamualaikum and a very good morning to all the panels and friends. My name is Nurul Izzah, and for my final year project, I am doing a review report on Mycosporine-like amino acids profile from Cyanobacteria. I have been supervised by Puan Arfida binti Rasul. Okay, firstly, what is Cyanobacteria and what is the relation with Mycosporine-like amino acids? Cyanobacteria is also known as blue-green algae, is a photosynthetic bacteria, which means it uses the energy from sunlight to produce and release oxygen gas into the atmosphere. It is believed to exist about 200 million years ago and is what responsible for the oxygenic environment we live in today. On the other hand, Mycosporine-like amino acids, or in short, the MAAs, is one of the secondary metabolites that is produced by cyanobacteria. The major function of this compound is to give photoprotection effect, which it absorbs ultraviolet radiation from the sun in various maximum range of wavelength. Due to this ability, it has been an important ingredient in sunscreen products. Here are all the research questions that I will be answering throughout my presentation. The first one is, does cyanobacteria produce MAAs as much as other MAAs producer? And what gap does this review cover in the knowledge of cyanobacteria's MAAs? Which order of cyanobacteria produces more MAAs? What is the most popular type of MAAs extracted from cyanobacteria? And what is the suggestion for further improvement in MAA bioprospecting? Okay, the problem statement arise from this study is that the types of MAAs found specifically in cyanobacteria is limited and not diversified. Secondly, they are rarely been commercialized, unlike the MAAs extracted from algae. Hence, the study is aimed to identify all the types of MAAs found in cyanobacteria and to examine the excellent properties of the MAAs and whether or not it is encouraged to be further commercialized in the industries. The significance of study is to compile the types of MAAs produced by cyanobacteria in order to see the most MAAs produced by cyanobacteria. Next, let's go into the content. The first one is cyanobacteria structure, types, and functions. So from the picture I have attached here in the slide, morphologically, cyanobacteria can be unicellular or filamentous and have a spherical, rod, and spiral shapes. The secondary metabolites are often produced by cyanobacteria in response to biotic or abiotic stress from the surrounding environment. It is to provide for protection and aid for the survival of the cyanobacteria. So the production of the secondary metabolites arise from the intracellular intermediates such as the amino acids, sugars, the fatty acids, which are then will condense into more complex structures by defined biochemical pathway. Cyanobacteria consists of 6 orders, which is Chlorella, Globacterialis, Nostocalis, Oscillatoria, Pleurocephalus, and Stegonometalis. Next is the MAAs profile from various species of cyanobacteria. Here, I attach the table that I have constructed showing all the types of MAAs compound extracted from 4 orders of cyanobacteria, which is Chlorella, Oscillatoria, Nostocalis, and Stegonometalis. So from the table, it can be seen that there are multiple types of MAAs that were extracted from different cyanobacteria. I took 3 types of MAAs that is common, which is Chinorine, Porphyra 234, and Mycosporine-like, to be further elaborate on its properties later. However, from a database, there are approximately 74 compounds that have been identified from various kinds of MAAs producing organisms, such as algae, coral, lichen, and some terrestrial animals. But from the table, there are only 31 types of MAAs that is produced by the 4 orders of cyanobacteria. Next is discussion. From the table before, Nostocalis order is the most cyanobacteria producing MAAs, followed by Oscillatoria, Protocalis, and Stegonometalis with 58%, 26%, 14%, and 2% respectively. It is also identified that the most popular type of MAAs found is Chinorine. Chinorine is reported to be produced by 16 species from Nostocalis order. Moving on to the Mycosporine-like amino acids properties. So in order for the cyanobacteria to protect itself from ultraviolet radiation, they produce the MAAs compound. So basically, these MAAs are produced by the induction of the ultraviolet radiation. They are actually a small water-soluble compound that is made up of cyclohexanone or cyclohexanine chromophores, which weight from less than 400 Dalton. And it possesses a very high molar extinction coefficient, which is the measurement of how strong it can absorb the light. And as you can see, it is 28,000 to 50,000 liters per mole unit. So these MAAs compound are able to absorb the UV radiation at different maximum absorption range, depends on its structure and characteristics. They will absorb the radiation and dissipate it in the form of heat energy. The MAAs are multifunctional metabolites that could also protect against free radical damage and boost cellular tolerance to desiccation, hypersalinity, and heat stress. Why is it worthwhile? Because in a study, the most group found from cyanobacteria, which is the Mycosporines group, have a maximum absorption between 310 nanometer to 362 nanometers. Next, let's look further into the profile of the common MAAs found in cyanobacteria, which is Chinorine. So Chinorine is one of the most abundant MAAs that can be found in various types of cyanobacteria species. The structure of Chinorine is by the condensation of the amino group and the keto group, and the replacement on glycine amino acid to hydrogen at position 3 of the cyclohexanone moiety. In a study, it has been reported that the production of Chinorine increased in cells exposed to photosynthetically active radiation plus UVB plus UVA rather than the cells that only exposed to photosynthetically active radiation plus UVA in Anabena variabilis. And it is also found that the Chinorine from Glycapsa species could absorb 333 nanometer radiation of UV light. Next is Porphyra 334. This one is a bit special because this MAA can withstand an extreme pH condition, which is in acidic aqueous solution. In a study, Porphyra 234 could absorb 332 maximum UV light in a pH 3 solution. Furthermore, Porphyra 234 that is extracted from Cyanobacteria AFA can absorb a maximum wavelength of UVA radiation at 334 nanometer and been commercialized in both sunscreen products. Last but not least is Mycosporine-like. Mycosporine-like possess a glycine moiety on carbon 3 of the cyclohexanine ring and a second amino acid, amino alcohol, or an amino system that attach to carbon 1. Having a maximum absorption at 310 nanometer, this compound is considered as an aminoketones that contain a usual cyclohexanone ring system linked to an amino acid. So in conclusion, MAs extracted from cyanobacteria has the potential to be further biotechnologically studied and commercialized more in various industries, such as the cosmetic products industry, the pharmaceuticals, agriculture, and many more. The highest order producing MAs is Cellulase, and the three most found MAs from cyanobacteria is Chinorine, Porphyra 234, and Mycosporine-like. My suggestion is that the methods in culturing cyanobacteria species could be diversified more so that new discoveries can be obtained. Here are the references for my review report. And so by this, we have come to the end of my presentation. Thank you very much for listening.
Nurul Izzah.
Yes.
Okay. Just now, you talk about this cyanobacteria, right? So right now, how they culture the cyanobacteria and then extract the MAs?
The methods in okay. So firstly, they will cultivate the sample. They will obtain the sample from mostly from aquatic organism, from aquatic environment, and then they will cultivate it. They will culture it with using several solutions such as some types of alcohol solutions, and then they will expose the culture to UV radiation so that the MAs will produce from the cyanobacteria, and then they will extract the MAAs, and then they will undergo the detection process and the characterization of the MAAs.
So the extraction process is what is the step extraction?
The steps for the extraction is
of the MAAs. Okay, never mind. All right. So you in your review, okay, between the four, right? What if Pyrophora 334, Mycosporine glycine, all these, right?
Yes.
Okay. So which one is the best? What are the best benefits?
The Chinorine MAAs because it has the highest molar extinction coefficient.
It has the
highest molar coefficient. Molar extinction coefficient, which is around 44,668 liter per mole.
Okay, Nurul Izzah, thank you. Time's up.
Thank you.
Okay, so we will proceed with the second last presenter, Siti Hajar binti Salim.
Can you hear me, Doctor?
Yes.
Okay. I will start the presentation now.
Okay.
Assalamualaikum and a very good afternoon to all panels and my fellow friends. I'm Siti Hajar binti Salim from AS20168 and my research topic is on the development and user acceptance survey of Chlorella vulgaris honey sunscreen under the supervision of Dr. Khairul Asfarazun. Without further ado, let's start the presentation. First of all, what is microalgae? Microalgae is a photosynthetic unicellular autotrophic organism with unique properties. It has high levels of polysaccharides and antioxidants, which are very important in biotechnology. The use of microalgae in biotechnology include in pharmaceutical, nutraceutical, and dietary supplements. Despite its high potential of source, there are less studies about the consequences of addition microalgae polysaccharide in sunscreen products and its effect on skin protection. Hence, this study is proposed to provide additional research-based data in this perspective. The study of microalgae composition and characteristics will contribute to a better comprehension and attention towards the potential uses of C. vulgaris bioproducts. It could also prove the endless benefits of C. vulgaris in a sunscreen and the effect it gives to the skin. The first objectives of this study is to optimize the Chlorella vulgaris powder, sunscreen and honey ratio via the box Benken design. Next, to conduct the user acceptance survey of the Chlorella honey sunscreen comprising of appearance, odor and texture on 20 participants. Moving on to literature review, C. vulgaris is a unicellular green microalgae that is widely found in freshwater, ocean, and terrestrial environments. For the past few years, C. vulgaris has become
Your
very
slide
famous.
is not moving. Are you the next slide or the
Yes.
first slide?
I'm on the literature review right now, Doctor.
Because here is the title.
Something wrong with your slide. Your slide not moving.
Should I stop sharing and do it again, Doctor?
No, you just press number 2, slide number 2, and then you have to on the right side, on the left side, there is a small screen, right? You just press.
What about now, Doctor?
Cannot see. Still cannot. It's not moving.
Still on the same slide. Slide for the first slide. Please move your cursor to slide number 2.
Yeah, I've already done it.
No, I cannot see your
Still not
cursor.
I think you have to stop and then share smaller.
Okay.
Yes.
Okay, Doctor.
Can you try and move it? Tengok. Is it working? Are you moving to the next slide now?
Yes. On literature review.
Still
Sunscreen.
Still it is on slide number 5. Can you go to slide number 6? Okay, alright. Okay, dah. Dah. You can.
Okay, boleh.
You can continue.
Okay. Should I just do it in the editing mode or presentation mode?
What's best?
Because I think if I use the presentation mode, it's not moving.
Okay.
You can pause it with the editing mode.
Okay, Doctor. Okay. Okay, so to continue, for the past few years, Chlorella vulgaris has become very famous in the cosmetic world. It is because C. vulgaris is very rich in carotenoids, which is a strong anti-aging agent, and also rich in amino acid, which is an important element of collagen to maintain the skin elasticity. C. vulgaris is also able to increase skin firmness. Thus, it can reduce stretch marks due to pregnancy or sudden weight gain or loss. Sunscreen is a type of an external wear that functions in protecting the skin from UV radiation that can cause sunburn or skin cancer. Most sunscreen usually consists of active ingredients that has the ability to block UV radiation. The last ingredients used in the sunscreen is honey, which consists of several vitamins. And the purpose of using honey for this research is to maintain the skin moisture. And the design of experimental use is the box Benken design, which include response surface methodology design, and it requires 3 different levels. Moving on to methodology. Firstly, we have to cultivate and produce Chlorella vulgaris. The C. vulgaris was grown in photobioreactor with Chlorella optimal media. The powder was also produced using the same method. This is the preparation of C. vulgaris honey sunscreen using the box Benken design. As shown in the table, there are 3 factors which are Chlorella, honey, and sunscreen. And each of these factors consists of 3 different levels. -1 indicates the lowest amount, 0 the medium amount, and +1 the highest amount. To produce the C. vulgaris honey sunscreen, first, the Chlorella powder was mixed with 10 ml of water to produce a homogeneous solution. And then this solution was then supplied at 3 different levels, 0.25 ml, 0.5 ml, and 1.0 ml. The sunscreen was also added at 3 different levels. The same goes to honey, which functions to add moisture to the texture. After mixing all 3 ingredients, 15 samples were then produced. And these 15 samples were then given to a total of 20 respondents to test the samples and answer the survey through a Google form. This survey consists of appearance, odor, and texture of the sunscreen. And the statistical analysis used are box Benken design, response surface analysis, and response optimization. Moving on to results and discussion, this table shows the number of responses that score between 7 and 9. As we can see, there are samples that receive relatively high score, such as sample 1 and 3, and there are also samples that receive low score, such as sample 4 and 11. This is the response surface analysis of appearance. There are 3 interactions. First, between Chlorella and honey. Second, Chlorella and sunscreen. And the third one is between honey and sunscreen. For the first interaction, both Chlorella and honey were preferred at the lowest amount. Meanwhile, for the second interaction, Chlorella at -1 and sunscreen at +1, the highest level. For the third interaction, both sunscreen and honey were preferred at the highest level, +1. As for odor, the first interaction, Chlorella at -1, honey at +1. Meanwhile, for the second one, Chlorella again at the lowest value, and sunscreen were preferred at the highest value. As for the third interaction, sunscreen was preferred at +1 and honey at -1, the lowest value. As for texture, for all interactions, both Chlorella and honey were preferred at the lowest value, meanwhile sunscreen at the highest value. And for the ANOVA test result, the shows that all factors for texture were insignificant to each other with a p-value of 0.05. This is the response optimization plot to show the most optimal value for each factors. And the maximum response will obtain when Chlorella and honey at the lowest amount, -1, 0.25 ml and 1.0 gram respectively, meanwhile sunscreen at the highest level, +1, 20 milliliter. The composite desirability indicates that 97.19% of respondents will accept the formulation at the said optimal amount. All in all, to conclude, samples with smaller amount of C. vulgaris powder were preferred compared to samples with larger amount of powder. Due to insufficient amount of data, interaction between all factors are considered weak. To solve this problem, first, the number of formulation should be increased to at least 1,000 grams of formulation. And the number of respondents should also be increased, as in this study, only 20 respondents were included and it was mainly consists of Malay females. Hence, the demographic population should be expanded so that we can study the effect of this sunscreen on various group of people from different races and age of group. With that, I end my presentation. Thank you so much.
Siti Hajar.
Yes, doctor.
Your respondent, how many? 20 also?
20 respondents, doctor.
What's the difficulties to get the feedback from respondent? Is it you send to only to 20 people, or you send more but they did not reply?
I sent to 20 people because it is because due to certain restrictions, such as this research was on the MCO time, so it was hard to collect samples more than respondents more than 20 people.
So how did you send the sample to them? Go to their house or?
Yeah, my neighbors, my relatives, and my friends that live around here.
I see. Okay.
The respondents are only female.
Was mainly consist of Malay females. Only two are Malay males, and there are no other races included in this study.
Oh.
Okay. In your conclusion, you said that they preferred this smaller
Samples?
amount of chlorophyll, right?
Chlorella vulgaris powder.
Yeah. Why do you think?
It's because we measured three parameters, right? Appearance, odor, and texture. So Chlorella vulgaris has a green color that is not very suitable for sunscreen. So that's why people usually prefer it when the amount of Chlorella is lesser or in small amount. The same goes to the odor because the Chlorella vulgaris has a strong, not very pleasant odor.
Because of the odor and and the
odor
color.
and appearance. Yep.
Okay. Okay, time's up, eh?
Okay, thank you, Doctor.
Come.
Okay, so we proceed with the last students, last presenter, Rose Nuzwi Natira.
Can you hear my voice, Doctor?
Yes.
Hello?
I can hear.
Yes.
I can hear clearly.
Okay. Assalamualaikum and hi, my name is Ruznu Zunatira binti Rosli. I'm from AS201-683. So today I will present my final year project entitled The Behaviour of Macaca, Fascicularis and Macaca nemasrina in Lumut Mangrove Forest. So I start with the background of the study. Long-tail macaque and pigtail macaques are very common and familiar amongst Malaysian people. Both can be found majority in lowland forest, peat swamp forest, and as well as in mangrove forest. Mangrove are marginal habitats for several terrestrial mammals. Obligate mangrove specialists among global primates are known to use this habitat facultative. Macaques usually live in ranked group and characterized by pattern of behavior such as agonistic display and social play. The problem statement of the study is there are no publication has been found on the study investigating behavior of both macaque species in the study area. It is also unsure if living closely in one area may affect the behavior within each species. There are two significant of the study. First is the studies of primates behavior provide important visions into our understanding of evolution and adaptation. The second one is it is also help in understanding more about the behavior of these two species in details as they are cohabitating in the same area. There are two objectives of this study. First is to determine the behavior of each species of macaque. The second one is to determine the differences in behavior displayed between different sex, age classes, and observation time in both macaque in Taman Paya Bakau, Bandar. As for the study area, the study was conducted in Taman Paya Bakau, Bandar in Sultan Manjung and owned by NGO, Kumpulan Aktivis Sahabat Alam, which is Kuasa. This mangrove park has different wetlands, which is river, mangrove island, and mud flats. The study animal is two, which is long-tail macaques and pigtail macaques. As for the data collection, preliminary observation was conducted for 3 consecutive days. Direct observation using scan sampling method within 2 hours of both macaques. So the total observation is 28, which is 10 observation in the morning, 18 observation in the afternoon. The observation done mostly in the main entrance section and observed according to the age class, which is adult, mother, juvenile, and infant. I used the SPSS version 22 and the test was one-way ANOVA test, and Kruskal-Wallis H test. We go to the result and discussion. As mentioned in the first objective, the most activity done by Macaca fascicularis during the observation is idle, which is the highest percentage. They are considered idle when sitting down and doing nothing. This macaque could be sitting, leaning their bodies on the tree trunk and lying on the front on tree branches with hands supporting their head. The second highest, which share the same percent, is are foraging and locomotion. The long-tail macaques do not usually rest too long in the daytime because they will try very hard to find food to survive at night. As for the locomotion, the long-tail macaques are diurnal animals that is active during the day, where they use one full day to travel from one area to another for searching the foods. The most activities done by Macaca nemasrina during the observation is idle, which is also the highest percent. During the observation, Macaca nemasrina did not select their resting site, which is idle at that time randomly, but selected a different type of sleeping site structure. Type of idle site structure. They seem to favor site with two canopy layer, a low and dense understory layer with many stems, and a second taller main canopy layer. The second highest is locomotion, which is 22%, as pigtail macaques are ground forager. They divided into small groups while foraging but keep in contact with the other groups through vocalization and range widely when searching for food. The two behavior that share the same percentage, which is 6%, are grooming and interaction with baby. In some occasion, an adult male pigtail macaque was observed to groom a mother carrying an infant. In this study, an adult pigtail macaque male was seen grooming an adult infant and vice versa. This behavior could be initiation of the male to attract the female. As mentioned in the second objective, the table, this is the comparison of age and behavior of Macaca fascicularis. The data is statistically significant. Post hoc tests show different p-value and mean between the ages. Overall, juvenile is significantly different between the ages. From the post hoc test, juvenile macaques foraging significantly higher compared to the other ages of the macaque. Next, we move to the interaction with baby. So the data is statistically significant. Overall, mother is significantly different between the ages. From the post hoc test, mother macaques interaction significantly higher compared to the other ages of the macaques. Next, idle behavior. The data is statistically significant. Adult is significantly different between the ages. From the post hoc test, adult macaque idle significantly higher compared to other ages of the juvenile. Next, comparison of sexes and behavior of Macaca fascicularis. So the data is statistically significant. So the graph shows different mean between the sexes. Overall, female is significantly different between the sexes. From the graph, female macaques grooming significantly higher compared to the male macaques. For the interaction with baby, the data is statistically significant. Overall, female is significantly different between the sexes. From the graph, female macaques interaction with baby significantly higher compared to the male macaques. The last one is locomotion. The data is statistically significant also. The graph show different mean between the sexes. From the graph, overall, male is significantly different between the sexes. From the graph, male macaques locomotion significantly higher compared to the male macaques. Next, the comparison between age and behavior of Macaca nemasrina. A Kruskal-Wallis test shows that the interaction with baby is statistically significant different in behavior between the different age class. So the data is statistically significant, and the graph shows different mean between the ages. From the graph, mother Macaca nemasrina have interact with baby significantly higher compared to the other of the age of the macaque. Infants are totally dependent on their mother for the year of life, for the first year of life, and during the first month, the young will rarely, if ever, break contact with the mother. Next, the comparison between sexes and behavior of Macaca nemasrina. So the data is statistically significant, and the graph show different means between the sexes. From the graph, female pigtail macaques have interact with baby significantly higher compared to the male of the macaque. We can see that mean of male are not far behind the mean of female, because during the observation there are also several interaction of the baby with the male. But Mr. Perry has stated that pigtail macaque society tend to slightly bias toward female, as infants stay with the mother for a longer time period, nurse for longer, and taking more of the female's energy. We move to the conclusion. So either locomotion and foraging are the most common behavior of the both macaque, which is Macaca fascicularis and Macaca nemasrina. The second conclusion is there are differences in behavior displayed between different sex and age classes in both macaque in Taman Paya Bakau Bandar. As for the recommendation, the data need to be collected more to get more significant result. I need to choose the balanced number of both species to obtain the strong interaction between sex, ages, and behavior. The last one is do more research that focus on sleeping behavior, as there are two species that cohabitating the same habitat that may have different sleeping site. That's all from this is the references. That's all from me. Thank you.
Okay, thank you, Rose Nuzwi. May I know how many times you went to Perak for data collection?
Went to Perak?
Your site. You said data collection.
I was in Perak. The study site with my home is 5 minutes only.
I see. So how many times you went to the site for a data collection?
Actually, I went to the study site 21 days, the total, but 3 days are the markups are not available. Maybe they go for the foraging at certain place, but I cannot collect the data for 3 days. The remaining days there is a data, even one markups only for one species.
Only once, Only many days, only one sample.
Certain day I have only one sample. Certain day I have 30 and above sample.
But the one sample you also counted in your results, is it? No,
yes.
not counted.
Counted.
How many samples are there? The number of animals involved in your study?
The Macaca fascicularis is 211. The Macaca nemasrina is 69.
Quite a lot, huh?
Yes, because Macaca nemasrina, there is not usually in the study site, but a lot of Macaca fascicularis in there.
Actually, for a good result, the one animal, the one samples only that you observe in one day, that one you should get reads out, not counted in your sample sepatutnya lah. Okay, other question, panel?
Okay, you have choose 2 species, right?
Yes, doctor.
So from the experiment, right? It's from the observation, right? Among these 2 species, is there any difference? What are the difference in terms of the behavior?
In terms of the behavior, Macaca fascicularis is more active than Macaca nemasrina, because of the size of the body, or maybe the Macaca fascicularis is more attracted to the visitors. But the behavior of the Macaca nemasrina is they like to stay at one place and stay silent.
Okay. Other than that, only that. One active, one silent.
Yes.
Other behavior.
Because I do not observe the comparison between these 2 species.
You observe their what? The
behavior.
Their behavior, right?
Yes.
So you should know, okay? Among these 2.
Only this one comparison that I can state.
All right.
Okay, time's up for Rose Nuzwe. So that's all. Thank you very much.
Okay. Thank you.
So we are now at the end of our sessions. 2 minutes past 1. So thank you very much to all lecturers, Associate Professor Dr. Noriza, Dr. Linawati, Ms. Azrina, and to all the presenters. Thank you very much.
Okay, welcome.
Thank you, Doctor.
Okay.
Thank you, doctors.
Okay.
Thank you, Doctor.
Okay. The student, I want to actually I want to discuss with Dr. Azani after this. The student can leave.
Student may leave the session now.
Thank you.
Okay.
All right, doctor.
Okay.
Thank you.
Okay.
Thank you all.